Look closely at a rapidly uncoiling fern. The physical coordination at the microscopic level requires highly specific instructions to execute correctly. But having millions of instructions is entirely useless without a reliable system to safely store, accurately read, and actively execute them in real time. The answer to this biological data problem is a specialized molecule called a nucleic acid. Nucleic acids have a single primary function. They are exclusively responsible for storing the code of life and transmitting genetic traits from one generation to the next. Biological survival relies entirely on the flawless management and communication of this hidden genetic data. To understand how this genetic information is organized, we must zoom in to its smallest repeating building block, the nucleotide. This diagram shows the chemical structure of a single nucleotide containing 3 parts, a 5-carbon sugar, a phosphate group, and a nitrogenous base. This simple module is life's universal data bit. They chain together, linking sugar to phosphate, to create massive sequences. The physical complexity of life on Earth derives entirely from the arrangement of this foundational module. The central, most secure archive of this genetic sequence is deoxyribonucleic acid, or DNA, which is locked safely away inside the nucleus of the cell. This model illustrates DNA's architecture, 2 nucleotide chains twisting into a double helix. The data is encoded using 4 bases: adenine, thymine, guanine, and cytosine. For stability, they follow strict pairing. Adenine connects with thymine and guanine with cytosine. DNA's locked dual-stranded structure makes it an incredibly stable hard drive for long-term data storage. But its immobility means it cannot actually build the organism itself. This creates a major biological bottleneck. The DNA holds the blueprints but is trapped in the nucleus vault, while the protein factories are located completely outside in the cytoplasm. Ribonucleic acid, or RNA, is the dynamic, flexible counterpart created to bypass this physical barrier. This compares their architecture. RNA is built as a highly flexible single strand, using a slightly different sugar called ribose. RNA also features a crucial chemical swap. Thymine is removed and replaced by uracil, which pairs with adenine. RNA's single-strand design is a physical adaptation, granting it the mobility to easily leave the nucleus and interact directly with the rest of the cell. RNA operates as a highly coordinated 3-part relay team to translate the stored genetic data into physical proteins. First, messenger RNA copies the required code directly from the DNA vault. Second, transfer RNA reads that code and physically gathers matching amino acids. Finally, ribosomal RNA acts as a factory floor, permanently snapping those gathered amino acids into a protein chain. This 3-part relay system allows the master code to be used constantly to build proteins without ever risking the original DNA file. This table outlines the clear functional duality between these 2 systems. DNA uses a double strand, deoxyribose and thymine, for storage. RNA relies on a single strand, ribose and uracil, for execution. A cell requires this precise division of labor between storage and execution to survive. DNA acts as the grand instruction book of life, carefully protected and preserved in the vault. RNA operates as the active messenger, the dynamic builder that reads those pages and translates the code into physical structures.